Hey everybody, I'm Brooks,
I'm Miranda.
and our protein that we'll be talking about today is collagen, specifically the type 1 alpha 1 chain.
So, when looking at the location of COL1A1, it is found in the subcellular level in the skin, tendons, and organs, and it is the main component of organic parts of the bone. In humans, 44 genes are present that encode for collagen. After it is encoded in the collagen, it is presented on the surface of the ER, where it is translated with the assistance of ribosomes. Next, the collagen moves inside of the ER, where it will undergo modifications. Lastly, the collagen will undergo final modifications in the Golgi apparatus before it is secreted into the extracellular space.
So collagen is a major protein in this extracellular matrix, and it's the most abundant protein in vertebrates. And collagen 1A1 is specific to humans and animals. It is not in plants, as collagen is a crucial protein or a crucial structural protein in multicellular tissues.
So, when looking at the normal protein function of COL1A1, it is a fibrous protein that has an elongated structure. It is composed of the triple helix fibers that we can see right here on our model, and it is strong, rope-like, and insoluble in water. Due to these characteristics, type 1 collagen provides structural support in bones, tendons, skin, and connective tissues. When looking at the specific function of this protein, it is the most abundant collagen in the human body. It provides structural support and strength to various tissues by forming the triple helix structure. It is found in connective tissues such as the bones, skin, tendons, ligaments, cornea, blood vessels, and it contributes to the strength and elasticity and structural integrity. It often works alongside other structural proteins, including type 3 collagen and type 5 collagen, to balance the strength and elasticity. Through these interactions, COL1A1 plays a vital role in maintaining the structural integrity and function of various tissues throughout the body.
This is the model of our mutated collagen. The mutation in collagen 1A1 affects type 1 collagen, causing there to be a couple hindering effects. The mutation causes Caffey disease, which is known as infantile cortical hyperostosis, which is a rare condition that affects various skeletal elements and connective tissues. So the collagen 1A1 mutation alters type 1 collagen by substituting arginine with cysteine. This disrupts the helical structure of the collagen fibrils, and the structural change affects collagen's ability to provide mechanical strength and stability to connective tissues. This leads to abnormal bone formation, soft tissue swelling, and inflammatory responses due to the impaired interactions with other extracellular matrix components as seen in Caffey's disease.
When moving on to normal amino acid characterization, we see that there is glycine and proline that are present and that are the main functional groups. We can see that represented by these two paper clips in our model. In addition, arginine contributes to stabilizing the function of the protein, particularly in collagen-related structures. This functional group plays a crucial role in the basic nature of arginine, allowing it to participate in hydrogen bonding and ionic interactions with other molecules. The charge of arginine guanidinium group helps stabilize protein structures, contributing to the overall stability of collagen and other proteins. So now we move on to hydroxylproline, which is a modified form of proline that has the additional function group of the hydroxyl group. So hydroproline further stabilizes the triple helix through hydrogen bondings between strands, which is represented through the blue ribbons at the end of our protein. Some properties of the original amino acid side chains includes that amino acids in COL1A1 are polar, therefore they are hydrophobic. Amino acids are neutral with a pH of 7. The exceptions are lysine and hydrolysine, which are both positively charged, and compared to the other 19 amino acids, the original side chain is small, therefore allowing for the triple helix structure. The types of interactions that the original amino acid can participate in includes hydrogen bonding between hydroxylysine and hydroxylized proline that stabilizes collagen structure. Ionic bonding and cross-linking with lysine and hydroxylysine enhances tensile strength. Hydrophobic interactions with proline contribute to helix formation. In addition to these interactions, glycine's small size allows for tight packing, ensuring the helix forms correctly.
So the main functional group that is present in the mutated side chain is the thiol group, which is found in cysteine. So in Caffey disease, the mutation alters a protein building block by replacing the amino acid arginine, which you can see by these paper clips in the original model, with cysteine, which you can see the mutations here in the paper clips, at protein position 836. So some properties of the new mutated chain is that cysteine is polar due to the new thiol group. Therefore, cysteine will be hydrophilic. Cysteine is neutral, has a neutral pH, whereas arginine is positively charged. So this mutation results in a loss of a positive charge. And then the mutation replaces the large positively charged arginine side chain with a much smaller neutral cysteine side chain. So it's going to be a more neutral pH and smaller. So one interaction that may occur with the newly mutated amino acid side chain is that if the amino acid is polar, it can form hydrogen bonds with nearby water molecules. Another interaction that can happen is if the new amino acid is charged, it can participate in ionic bonding with oppositely charged residues. And the mutation also results in a loss of flexibility, as you can see by the cracks in the mutated model. And the increase in size will cause the triple helix to be less stable and more resistant to the surrounding water molecules. So mutation affecting the triple helix can lead to improper folding or destabilization. This may cause some of the mutated regions to produce defective collagen, which can disrupt the normal collagen. So the misfolded proteins in the cell can cause stress on the ER, and abnormal collagen secretion can weaken the extracellular matrix. This reduces stability and impairs cell signaling. So the organism can experience skeletal, connective tissue, and vascular issues due to the mutations. The brittle bone disease, weaker ligaments, and an increased risk of aneurysms are just a few examples. Collagen-related diseases can arise from genetic defects or nutritional deficiencies that affect post-translational modification, assembly, and secretion, among other processes involved in collagen production.
So when comparing our normal model versus our mutated model, we can see that the prime difference is the amino acid groups that are present, as we can see here by this paperclip compared to this paperclip. We can also see that in our mutated model that there is tension put onto the triple helix that impacts the function of this collagen.
So for the primary structure of collagen, it is just a polypeptide chain. For the secondary structure, this is when the chain forms an alpha helix structure. The tertiary structure is when the polypeptide chain folds and creates a three-dimensional shape. And then finally, in the quaternary structure is when the three polypeptide chains come together to form the triple helix.
So when looking at the limitations of our project, it is difficult to represent the contributions of amino acids in using candy, such as glycine, that tightly packs together the triple helix. It is also difficult to scale the mutations, and it is difficult to show the chemical changes that are happening, as we only showed the physical changes. Thank you so much for listening.
Thanks for listening.